Conclusion: 2,5-diketo-D-gluconic acid reductase A will be the therapeutic gene of interest in our study, and we would like to insert it into the RAAV vector. After completion of triple transfection, RAAV variant particles containing the 2,5-diketo-D-gluconic acid reductase A gene in the DNA and a Rep2/Cap2 capsid are created. Thereon, we will inject into the bloodstream, intravenous or interarterial or intramuscular, this recombinant adeno-associated virus into the central nervous system of a HIV/AIDS patient and wait for integration of the virus carrying the therapeutic gene of interest onto the specific site in human chromosome 19. The 2,5-diketo-D-gluconic acid reductase A gene will be expressed from human chromosome 19. There will be an increase in the cellular level of 2,5-diketo-D-gluconic acid reductase A protein, and we would expect an inhibition on HIV-1 integrase. This will prevent the chelation and formation of hydroxyl groups on the 3' ends of the viral DNA, and also prevent ligation and integration of viral DNA into the host genome, that is the host chromosome, thereby affecting the infectivity of the HIV virus. These steps will be beneficial in the treatment of HIV/AIDS at the nuclear DNA, that is chromosome level, since RAAV effectively block the ability of enzyme integrase to perform its action in assisting HIV nucleic acid delivery into the cell nucleus. On another note, the field of biological engineering has seen significant growth in recent decades. Within this field are devices called biosensors. A biosensor is an analytical device for the detection of an analyte that combines a biological component with a physico-chemical detector. In our case, we would like to develop a AAV technology-based biosensor for measuring the levels of therapeutic protein, gene of interest, expressed in blood using a protein probe. This will indicate when it is necessary to take additional RAAV infusions and be important in determining the appropriate viral dosage requirements and dosage timings. At least one research group has designed a sophisticated multifunctional probe that is built around a 13 nanometer gold nanoparticle. The nanoparticles are coated with hydrophilic oligonucleotides containing a Raman dye at one end and terminally capped with a small molecule recognition element, example biotin. Moreover, this molecule is catalytically active and will be coated with silver in the solution of AGI and hydroquinone. After the probe is attached to a small molecule or an antigen it is designed to detect, the substrate is exposed to silver and hydroquinone solution. A silver plating is happening close to the Raman dye, which allows for dye signature detection with a standard Raman microscope. Apart from being able to recognize small molecules, this probe can be modified to contain antibodies on the surface to recognize proteins. When tested in the protein array format against both small molecules and proteins, the probe has shown no cross-reactivity. In conclusion, to learn about my background and humble beginnings in life and past work, please read my biography posted on the Nobel Prize Foundation website. Scientific references/citations can be found in the reference list section at http://www.DoddiNobelLecturePresentationVideoPart6.blogspot.com. For an original lecture transcript and resume, please email me at sdoddi11@hotmail.com. Thank you for your nominating me for the Nobel Prize, and an extended thank you also to Nobel laureates in medicine, Dr. Louis J. Ignarro and Dr. Richard J. Roberts, for their patience.